Thank you. Thanks to you guys. Do you hear me well? Yeah?
Yeah.
Okay. Okay. So, well, thanks for the kind invitation. Thanks for Francesca to showing us how bone marrow-derived macrophages work. And what I'm going to show you today more is how those tissue-resident macrophages, the macrophages that are the first encounters that when a tumor cell emerges from the tissue and how these cells work. I'm going to get it into a little of bone marrow-derived max, but I think it's the work of Francesca and many others before have really shown how these cells operate. So I'm gonna focus on another flavor of max. So as you might know, macrophages were described many years ago by Elin Mednikov, in which using a very rudimentary microscope, he observed that these cells are highly phagocytic cells, right? So that's their main function. And depending on the food they eat, they can trigger either an immunosuppressive or an immune modulatory transcriptional program, as you can see here, exemplified by alveolar macrophages when they eat debris uptaken from cancer cells in the lung particularly. And one of the features of these cells, and that really caught my attention while I was deciding where to go to do the postdoc, is that in a seminal work from the lab of Miriam Merat, they observed that depending on the tissue in which these macrophages reside, they can really trigger like very different transcriptional programs that then make these macrophages capable of develop like very tissue-specific functions. And this is amplified here by the expression of some of these transcription factors. And I'm going to focus, for example, in the lung, the expression of PPR gamma triggers the expression of GM-CSF receptors and also allows these cells to engulf and remove the surfactant in the lung. In the strike contrast, for example, when you look at tissue-resident macrophages in the peritoneal cavity, these macrophages express particular transcription factors such as GATA6 or RXR, which upon the signaling that they receive of retinoic acid really allows these cells to also phagocytose and to prime these cell responses in the gut. So it's really unique features that reside in the tissues, which allows the function of macrophages. And you can think of a tumor of a tissue as well that is developing. So those cells with different react to the different stimuli they are sensing close by. And one major difference about the macrophages that was also described in Miriam, but not in many others, is that depending on the tissue we are looking at, there are macrophages which really seed the tissue at very early stages during our embryonic hematopoiesis, versus another subset of macrophages, such as the one in which Francesca was focusing on, which arise from adult hematopoiesis, those progenitors that are going to differentiate and upon inflammation are going to sense that they need to go to a particular lymphocyte, so a tumor, and developing tumor, for example. So this is one of the differences which I will focus on today. So one of the things about this heterogeneity that also Francesca has pointed out is that macrophages function differently either in homeostasis or in inflammation, and that these macrophages are really plastic regarding to the factors they send in the tissue. And some of the emerging paradigms in which I will want to focus in the future is trying to understand how this heterogeneity is also determined by the interactions that these macrophages establish, the ontogeny, but also the different sources of heterogeneity, such as the energetics these macrophages utilize, the response to treatment, and the development and progression of disease and metastasis. But not only that, the innate cells, not only macrophages, learn a little as well, right? So it's not, we don't react the same when we have experienced aging, obesity, viral, bacterial infection. So those macrophages will also sense that previous diseases. So all these factors have driven not only immunologists, but also cancer researchers trying to target macrophages in solid tumors. And as Francesca also has exemplified, these macrophages are the largest immune compartment in solid tumors. They have been related to many pro-tumoral functions, but something that hasn't been looked in detail, in which I think that I will show you today, is that it is important that we pay attention in which tumor, in which organ the tumor develops, because, as I mentioned to you before, that is going to determine a different flavor and a different activity of these macrophages. As such, it's not only important the tissue, but also how this tumor progresses, right? Because the interactions that these macrophages are gonna immerse with different components of the tumor microenvironment are gonna vary a lot depending on the stage of the tumor. As you see, when precancerous lesions, almost normal lesion, as long as this tumor develops, when there is immune editing, there are many tolerogenic cells, such as, for example, Tregs, we were discussing with Eugenio before, that will come into place. And when all this inflammation and all these myeloid-derived suppressor cells came into place. So that's also an important concept that we as immunologists need to start thinking about. And most importantly is that most of the macrophage biology that has been uncovered till now is being based on in vitro studies. And with the advent of single-cell technologies and more and more potent flow cytometry techniques, what we have starting to see is that this M1 and M2 paradigm that Alberto Mantovani described in the 80s does not longer recapitulate what we see in vivo, right? So importantly also in light of this previous finding, this previous biology, which really set the stage for all of us, is that this tissue-resident compartment has not been put into the equation. So this heterogeneity also deserves a need of defining this lineage. So that's what I'm going to tell you today. So the project starts just, raising the hypothesis that these different ontogeny unrelated macrophages will play a different impact in modulated anti-tumor immunity. And we decided to focus on non-small cell lung cancer tumors, not only because it's one of the major cancers worldwide today, not only in the States, but worldwide. It's also because the lung allows us the perfect scenario in which we can test this hypothesis. So in the steady state now, while you are breathing, all of us, we have like a tissue-resident compartment, which arises from fetal liver progenitors that seed the tissue very early on during embryo development, but also cohabiting with this compartment of tissue-resident macrophages, we have bone marrow-derived macs that are gonna seed the tissue and reside in the interstitium, in the lung interstitium. So we decide to focus also into this question because we believe that trying to understand how tumors evolve on a tissue-specific manner can really help us to guide future treatments in targeting this population. So the models we utilized was two different models, so to speak. The first of it was an orthotopic model in which we transfer via tail vein lung adenocarcinoma cells that they express KRAS mutation, they are deleted for p53, and that we have engineered to express GFP so that we can follow longitudinally the growth of the tumor and also both the adaptive and the innate response. And concomitantly to this model, we also wanted to understand how this macrophage heterogeneity at the level of ontogenic macrophages was occurring in non-small cell lung cancers in early stage. So we decided to take an unbiased approach, utilizing single-cell RNA-seq, because as you know, macrophages are going to change quite a lot into a progression tumor. So we decided not to focus on bias gating strategies and try to analyze their transcriptome profile to have a complete picture of this macrophage heterogeneity. So I'm going to show you now the data that we gather both for human and for mice, and hope we can discuss on it later on. So when we analyze and we work with a super talented PhD student in Miriam's lab, Andrew Leader, what we observe is that when we start analyzing those tumor lesions, and when we analyze those macrophages, we observe four different main groups. As you can see here, this is a heat map that exemplifies the most differentially expressed genes that we encounter in these different clusters of macrophages. And for the sake of time, I'm going to focus only on this cluster 1 and cluster 2 that contains macrophage canonical genes that we were able to classify, and not into these other 2 clusters which correspond to inflammatory CD14 monocytes and CD16 monocytes. So as you can see here, just at a glance, these canonical genes that were the most detected and the most differentially expressed between the different clusters, none of them were corresponding to this classical M1 and M2 classification. Doesn't mean that they don't express, but they were not the top ones that we gathered in our single cell analysis. So then when we get into our mouse model, the first question we ask is, okay, do we have the same heterogeneity, or how does that work in mice? So what we observe is that, again, the same number of clusters, the same that were defined also in the literature, we're able to identify the same clusters, these inflammatory monocytes, these pathogenic monocytes, and we again find 2 different clusters that show kind of different intermediate levels of canonical monocyte and macrophage genes. So next we wanted to identify what is indeed the, one of the things that we wanted to also try to understand doing this cross-species analysis is trying to say, okay, we were able to identify the different groups of macrophages, but how can we bring into the community a consensus to utilize macrophage markers so that they can really identify this entire heterogeneity. So in order to do that, what we came up was with an approach that Andrew developed, in which we could really compare the transcriptome profile between the 2 species, between the genes that were preserved indeed among these 2 species. And as you can see here for this cluster 1, what we observe is that some of the genes that were preserved were MRC1, classical M2 marker, Sigle1, Marco, PPR gamma, both in mouse and in human. And the same holds true for this other subset of macrophages and for inflammatory monocytes and pathogenic monocytes. And if you are interested, you can just go and dig it into the list in the manuscript. But still, we didn't know what was the origin, right? We identified these populations, both in human, both in mice, but we didn't have any way to really say, okay, this is the same population. So in order to do that, we collaborate with Boris Reises, in which he developed a couple of years ago a model in which you can really trace all the cells we derive from bone marrow derived hematopoietic stem cells. So we utilize his mouse model, and in this mouse model, MAP17, what we did is upon injection of tamoxifen, we induced the recombination of the ROSA26 locus, and that allows the tracing of this HSC derived progeny. And importantly, any of these precursors, which are from fetal liver hematopoiesis, express at any given time this MAP17 gene, so allowing us to track a bona fide HSC-derived progeny. So we validate those results utilizing flow cytometry, and what we observe utilizing this mouse model is that both in the blood and in the lung, the vast majority of myeloid-derived cells, which are derived from bone marrow hematopoiesis, were efficiently recombined, but still there was a lineage of cells that did not show any bone marrow HSC-derived composition, very little. And this is what we call this tissue-resident macrophage compartment. We localize these cells, as you can see here, and the vast majority of these cells that express CD206 were not bone marrow derived. And this is consistent with the work of Martin Gilliam, Catherine Sawai, and also Simone Jona and Stephen Jones' lab, which they show already using another different model for tracing that if in the steady state, at least tissue-resident macrophages in the lung, which correspond to alveolar macrophages, do not arise from bone marrow-derived cells. So then the obvious question, well, okay, what does it happen in the tumor microenvironment? So we repeat the same experiment with this MAP17 model, but this time we utilize this CD11B and CD11C enriched myeloid cells in order to discriminate then and utilizing single-cell RNA-seq cells which were from embryonic origin versus cells that were from bone marrow-derived origin. And what it turns out, and it was quite surprising because it was somehow against what we were expecting is that in tumor-bearing mice, we observed that indeed there was still a resident compartment that showed barely contribution. So those bone marrow HSCs do not really repopulate the population of tissue-resident MACs, and we observed that the other contribution of macrophages and inflammatory monocytes and pathogenic monocytes were showing an enriched contribution of bone marrow-derived cells. Then, retrospectively, came into our modular analysis in which we analyzed those genes that were preserved between mice and human. And what we observed when we have very adult tumors, and this is consistent also with previous data, is that both in mice and in human, this tissue resident compartment is dramatically reduced, and that occurs at the cost of an expansion of these bone marrow-derived macrophages that we find in the tumor. So we have certain moments in which have really a dance of macrophages, and we have this tissue resident compartment and this bone marrow-derived compartment that really take over at later stages of tumor progression. So one of the cool things about single cell is that, okay, you get crazy among a lot of transcripts and months of analysis, but something that it's useful is that really they can allow us to identify markers that can really be used to challenge the biology, right? And that was our really motivation to do these experiments. So we utilize the single cell data to identify markers that can really be used as a bona fide markers of tissue resident MACs in the lab. And it turns out that when we combine both CGLEP1, also known as CD169, and MRC1, again, previously characterized M2 markers, what we observe is that utilizing the specific reporter mouse that we developed, is that the combination of these two markers allow us the identification of this tissue resident macrophage compartment in the tumor microenvironment. So next, we decide to challenge this model using, we decide to try to identify how this progression of macrophages was populating the tumor microenvironment. And I'm just showing here how this decrease occurs. And for that, what we did is we utilized a longitudinal experiment in which we injected these KPGFP cells, and then we follow over time how this tumor progresses. And as you can see, these tissue resident macrophages identified here by the expression of CD206, also known as MRC1, is that these tissue resident macrophages are the first cells that interact with the cancer cell, and that as long as this tumor progresses, these macrophages are somehow like spelled out. And when we quantify this distribution of macrophages, we observe that indeed these macrophages, the tissue resident compartment tends to be more located in the peritumoral areas. This was not an artifact of the model that we utilize, a metastatic model, but we also utilize this genetic engineer mouse model with the same mutations, KRAS and P53, generated by Tyler Jacks. What we observe is that also these tissue resident macrophages are the first macrophages that interact with the mutated cells, and that as long as this tumor progresses, we observe the same trend that these macrophages are somehow expelled out from the tumor foci. So, okay, this is what it occurs with the tissue resident compartment, but what does it happen with the MOMACs? So we utilize another lineage-traced model to really identify these bone marrow-derived macrophages utilizing CXCR1 3ER promoter, and upon tamoxifen diet, we are able to identify YFP positive cells. And what we observe is that when we quantify the distribution of these macrophages, we were not able to see a preferential location of these MOMACs compartments. So highlighting that these MOMACs really do not, when they arrive as monocytes, they are located till now in a random disposition. This is the quantification that we utilize for flow cytometry. As you can see, in ovarian tumors, this tissue resident compartment remains absent of the tumors wherever. However, this bone marrow-derived compartment is significantly expanded. So, well, one of the things about single cell is that it allows you to resolve heterogeneity, but it doesn't allow you then to dig get deeper into functional programs of macrophages. So for that, what we did is just we isolate the macrophages from these tumor lesions, and for that we utilize bulk RNA-seq to get a more comprehensive program of these cells in the tumor microenvironment. And we did that at very early stages because we reasoned that these cells, something when they are doing their effect, it might be early. And as you can see here by this heat map, what we observe is that these macrophages really start upregulating a program that is related to metalloendopeptidase activity, so remodeling activity, but also with class 2 activation, peptide antigen activity. And let me go, let me come back to this data later. So in order to try to mimic that transcriptional program in vitro, what we did is to establish 3D in vitro cultures with this KPGFP together with 2 different sources of macrophages, these tissue-resident macrophages that we isolate from the tumor lesions, from early tumor lesions, and these bone marrow-derived macrophages that we also isolate from later stage tumors in this case. And what was very cool to see is that when we develop an assay in which we can follow also over time how these organoids were growing, what we observe is that these the organoids that were cultured with the source of tissue-resident macrophages invade way more GFP area than when we compare it to the source of bone marrow-derived macrophages. So then we reached out Julia Aguirre-Viso, which is a magnificent cancer biologist that taught me many, many things about cancer. And we decided to set up an experiment in which we were really getting a closer look into these spheroids. And for this, it was very evident we didn't even need to know which were the slides we were looking at, but when we were looking always at organized co-culture with these tissue-resident macrophages, what we observed together with fantastic PhD student, Erika, is that they lose the expression of ICA variant, absolutely, in a very significant manner. And that was not the case when we were co-culturing this with these tumor bone marrow-derived macrophages. Interestingly, we were also be able to see that these organoids, they start like protruding kind of lamellipodia, like being showing more invasive features. And that was not the case when we were analyzing organoids culture with bone marrow-derived macrophages. And as I simplify here, I'm just showing here a video because imaging is always cool to see, but what you can see here, all these transparent cells hanging around, they are tissue-resident macrophages that imagine that they will be in your alveoli scavenging all your bacteria or all your COVID. But here, what they are scavenging are cancer cells, right? So you can see here how this kind of lamella is formed. It's going to form a more invasive spheroid, but also it was very interesting to see that single clones of tumor cells are taken by macrophages and they are not phagocytoids. This, when you follow them over time, they are going to develop a new spheroid. So that was super cool to see. And indeed, when we repeat this experiment with bone marrow-derived macrophages, what we observe is that this phenomena was not taking place, but the organoids were expanding dramatically. So I will tell you this in a couple of years, I hope. Okay, so one of the things is that, okay, this is what it happens with macrophages. You put macrophages in a dish with cancer cells, but does this kind of increased migration happens also in vivo? So in order to answer that question, we utilize CD169-DTR that, as I show you, in combination with CD206, efficiently takes all this population of tissue-resident macrophages. And what we did is challenging this system utilizing diphtheria toxin in order to get rid of these tissue-resident macrophages. So what we see is that when we utilize both tissue-resident macrophage sufficient or tissue-resident macrophage deficient mice, and when we analyze the cancer cells that we have transferred in these mice, what we observe is that very early on in day 5 lesions, what we observe is that these cancer cells indeed lose the expression of TWIS1 and CED1, which are canonical factors that are expressed canonical transcription factors, which are expressed when endothelial cells engage into a mesenchymal phase. So suggesting that these tissue-resident macrophages indeed in vivo promote epithelial mesenchymal transition. And then as a result of this phenotype that occurs at very early stages, what we observe is that when we deplete these macrophages early on and then we transfer these cancer cells, the tumor burden is dramatically impacted in this model. And as I told you before, one of the things that was really catching my attention is that these tissue-resident macrophages start to upregulate class 2 molecules as well as the recruitment of some T cell recruitment molecules. So I say, okay, maybe these cells are also like somehow priming a T cell response. So Andres told me well to look into the tissue, and I was very obsessed to trying to identify what were these macrophages interacting with at these early stages. And something that I was always seeing is that this kind of menage a trois in which these cancer cells establish close contacts with regulatory T cells, and that whenever you see this interaction, there is always like an alveolar macrophage hanging around there. And as you can see, these regulatory T cells are predictors of poor outcome response in not only in patients but also in our models. As you can see here, this is like this kind of healing formation that is observed with these tissue-resident macrophages recruited and regulatory T cells. So next, I said, okay, how this evolution of tissue-resident macrophages occur together with the regulatory T cells. So what we observe is that using regular wild type, we observe that very early on there is a massive expansion of regulatory T cells in situ in the tumors at very early stages. However, when we get rid of these tissue-resident macrophages, what we observe is there was a dramatic reduction of these regulatory T cells in the tumor microenvironment. Sorry, somebody's calling. And also these regulatory T cells somehow shift their phenotype. And we assess that by the expression of CD73, CTLA4, but also IL-10 expression in regulatory T cells. So somehow these tissue-resident macrophages, at the same time that they are speaking with these cancer cells and inducing this pro-migratory program, they are also modulating the expansion of regulatory T cells on their phenotype. So as a result of this, what we observe is that at very early stages, when we have depleted these tissue-resident macrophages, before we observe a dramatic reduction of regulatory T cells, and also a very infiltration of the tumors by CD4, by CD3 T cells, that then when we quantify it, actually what we observe is a massive infiltration of CD4. And as a result of this, we also observe a better effector, anti-tumor immunity effector response, as quantified here by the expression of TNF-alpha and interferon gamma CD8 T cells, and a better ratio of CD8 Tregs. Interestingly, when we deplete these macrophages later on, and if you remember the imaging, when this kind of healing of these tissue-resident macrophages is already formed, what we observe is that depletion in later stages does not have any result on tumor burden. And as such, we didn't see any kind of regression in regulatory T cell compartment, nor a better infiltration or effector response in the tumors. So I hope I didn't go too fast or too long in time, but what I show you today is just a summary of how the complexity of macrophages operate, and just looking at ontogeny. What we observe is that this tissue-resident compartment plays a major role at early stages, initiating remodeling and driving immunosuppression by regulatory T cells. Whereas this bone marrow-derived compartment, when we look at these tumor cells, and actually what we observe is that they really promote the expansion. They really give a boost of pro-mitotic cytokines into the tumors. I'll show you today also that these different ways of ontogeny existing macrophages accumulating these tumor lesions, and that the adult monocytes cannot give rise to these tissue-resident macrophages, but that might be only true for the lung. It could be completely different in other organs. So this is our work highlights really the importance of understanding macrophage biology on a tissue-specific manner. As I mentioned to you, these tissue-resident macrophages are mostly relevant to tumor inception, and the way we are trying to now identify ways to modulate this compartment is trying to activating them using very low doses for activating these macrophages, specifically in order to reduce their tolerogenic potential. And with that, the most important slide is the acknowledgements slide. And this project for sure couldn't have been taking place without an amazing mentor, amazing immunologist, an amazing woman, which is me and Merat. and two fantastic students, one fantastic student, Andrew Leader, which led all the computational work, who really guide me into single cell analysis, a fantastic lab technician, Jessica Leverishale, our collaborators, especially Effie Kenigsberg, which was also the mentor of Andrew in single cell analysis, and amazing cancer biologists such as Erika and Julia Guiso, which really were fundamental in this work, and the rest of our collaborators, the course, and of course the funding. So with that, I will be happy to take any question. Thank you.
Thank you, Maria. Thank you for this great talk. It was really a fantastic and amazing work. So if you have any questions, you could type it in the chat. In the meantime, I used to have one question when I was reading the paper, but now you already solved that. So I was thinking on, so we have one question from Paula Boss, and she says, congratulations to both of you and thanks. I have one quick question. Do you see decreased proliferation in the lung Treg cells, Maria?
Yes. I mean, we only see decreased proliferation when we perturb the tissue resident compartment. I did also some perturbation experiments utilizing CCR2 knockout mice, in which I also observed depletion reduction of Ki67 regulatory T cells, but I didn't see modulation in their profile. I didn't see changes in IL-10, CD39, CD37. So those are the things actually, Paula, I wanted to ask you. So.
Okay. Yeah, I was wondering, Maria, I don't know if it was expected to, if you did again the single cell RNA-seq analysis when you deplete these tissue resident macrophages. So what happened to the other myeloid compartment? Does maybe the tissue resident macrophages are somehow creating this microenvironment that reshapes all of the myeloid compartment there?
Yeah.
Did you
I mean, I would expect that, because what we observe is like having both compartments, and how also like the tissue resident macrophages are like trying to calm down the expansion, whereas really the bone marrow derived ones are the ones that foster it, like really promote massive expansion. So if I were to speculate and discuss about what would happen in a lung deployed completely of tissue resident macrophages, of course, we will see that tumors do not evolve as fast and do not induce this metastatic potential. But for sure, the tumors are still growing, right? Because there are other macrophages which are somehow want to try to feed the tumor for. So that would be what I would expect.
So would you say, going back to my question with Francesca, would you say that these tissue resident macrophages are maybe in crosstalk with the bone marrow to, after getting in contact with the tumor cell, and then maybe reshaping, giving signals to the bone marrow that?
I mean, they definitely, so like this peripheral control of macrophage tone, but I think it's not exclusive of macrophages, right? It might happen with neutrophils, but with T cells as well, or B cells, right? There's an organ when we age, when we get older, that masters and excels at controlling of all of our immune system, right? So in the moment there is deficiency of some reservoir, the machinery starts, right? So you somehow need to preserve the integrity of the organ, and the bone marrow controls that.
Yeah, okay, thank you. I don't know if we have more questions here. If not, thank you very much, Maria. It was a really fantastic talk. Congratulations on all your work. Congrats on your new lab.
Thank you.
Upcoming sessions today, next week, and we have an editor session. We can talk with editors from different journals, and a neuroimmunology talk as well. Thank you so much, and see you next week. Thank you, Maria. Thank you, Francesca. Congratulations.
Thank you very much for the invitation.
Bye.
Bye.
Thank you, Federico.